Good evening, Arya. Good evening, Samisht. Okay. So, I'm Rahul Saha, and we were discussing the glycolytic pathway in the last class. So, we had discussed till the payoff phase. Okay, we had discussed till the payoff phase beginning. Like I had started with the glucose becoming the glucose 6 phosphate, then it becoming the fructose 6 phosphate, then fructose 1,6 bisphosphate, and then becoming enzyme, you can say aldolase, converting it into, let's say, fructose 6 phos 1,6 bisphosphate was converted to by the enzyme aldolase to two specific triose phosphates like glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Acetone phosphate. So these two compounds, like we had talked about, and now dihydroxyacetone phosphate will be changed back to glyceraldehyde 3 phosphate by use of an isomerase enzyme. There will be an isomerase enzyme which will change dihydroxyacetone phosphate to glyceraldehyde 3 phosphate. 3 phosphate. So now if you see, this one is a 6 carbon compound, fructose 6 phosphate is a 6 carbon compound. Now dihydroxyacetone phosphate and glyceraldehyde 3 phosphate both are 3 carbon compounds. So now we have one, this one, we had one, this one, 3 plus 3, 6 is equal to 6. But this one, all the dihydroxyacetone phosphates has been converted or have been converted to glyceraldehyde 3 phosphate. So total you will be having 2 times glyceraldehyde 3 phosphate. So again 3 carbon into 2, you are getting again 6 carbon, which maintains the stoichiometry of 6 carbon, which is present in the, you can say, fructose 1,6 bisphosphate. So this is the thing that you will be needing to look at, right? After this, what happens is, now we enter the payoff phase. So till now I had explained to you the structure also. If you see the last video, part 1, I had explained the structure also. And you can say, I'll just redraw the structure of glyceraldehyde 3 phosphate for you. So glyceraldehyde 3 phosphate, it looks like this. You have CHOH, then COH, then you have CH2 this side. I can draw like this. Then you will be having O here, and then one phosphate group. P is here, CO4 3 minus. This is your glyceraldehyde 3 phosphate. Okay. Then if you want to check dihydroxyacetone phosphate, which is its isomer, okay, which is its isomer. So dihydroxyacetone phosphate starts with CH2OH. Okay. Then it will have a double bond. O, one hand will be here free as the valency of carbon is 4. Then again CH2. O, again this is the phosphate. So this is your dihydroxyacetone phosphate. So we had 2 phosphate groups in the fructose 1,6 bisphosphate, one at the first position, one at the 6th position. Now you have 2 phosphates here. Okay, so this will be converted to this one. So you'll be having 2 glyceraldehyde 3 phosphate molecules, 3 carbon each. You can say 1, 2, 3, 3 carbon each, and therefore 2 glyceraldehyde 3 phosphates means you will be having 2 phosphates also. So the entire stoichiometry of the reaction is completely conserved. Now after till now we have now completed the preparatory phase. Now we are over with the preparatory phase. Now we will be commencing the very important payoff phase. Now why I am calling it the payoff phase commencement is there is no cycle, right, where we will be only using up ATP and not getting anything proper. Other, like if you use up ATP, you are supposed to get some molecule, like you are supposed to get starch, or in case of plants, sucrose in case of plants, or fatty acid in case of animals, whereas glycogen, something you should be getting which can be stored as an energy reserve, okay, or you should be synthesizing nucleic acid, something worth, you can say, spending ATP on. Here, till now, we have only broken down glycolytic in the glycolytic pathway. The glucose still, you can say, 2-triose-phosphate glyceraldehyde 3-phosphate, which is of not great use. But we have already, if you see this reaction, if you have a look, we have already lost 2 ATP molecules which are quite crucial and extremely, you can say, worthy molecules because they are the energy currency. We have broken down the gamma phosphate bond and converted ATP to ADP and phosphorylated these compounds. Okay, so phosphorylation also why it is done, I had explained to you in the last class, so that the phosphates, they cannot come out of the cell. There is no transporter for carbohydrates which have been phosphorylated, so it is booked inside the cell, it is trapped inside the cell, and the ATP, after you break down the ATP, the gamma phosphate bond, that bond gets attached. Okay, that bond breakage, the energy is almost completely conserved after the formation of this phosphate bond or this phosphate group when it is attached covalently to this target compound like the carbohydrate, and also this phosphorylation. Okay, this phosphate group phosphorylation, that energy also can form various, you can say, covalent and non-covalent interaction with the enzyme's active site, and thus it can lower the, you can say, the activation energy of the reaction, which we had talked about in the biomolecule chapter. So that is why phosphorylation is done. So that also has a specific, you can say, reasoning that why phosphorylation is happening. But till now I haven't been, or there hasn't been any significant molecule synthesis. It's just breaking down glycolysis. You cannot store glyceraldehyde 3-phosphate, you cannot store fructose 1,6-bisphosphate. So the, you can say, the real suspense is being built up why 2 ATPs were, you can say, broken down. Why were they used up? So now you will get the answer in the payoff phase. You will see me getting, or the glycolysis getting back 4 ATP molecules. So the net of 2 ATP will be the, you can say, consequence of this glycolysis, because 2 ATP you have lost. Now you will generate 4 ATPs, so 4 minus 2, so net 2 ATPs you will be getting back. So how you will get back? Just look at this payoff phase now. Now you have 2 glyceraldehyde 3-phosphate. Okay. 2 glyceraldehyde 3-phosphate. Okay. After you get this 2 glyceraldehyde 3-phosphate, okay, what you have is, there is a specific enzyme now. This is a very, very special reaction. This is a specific enzyme known as glyceraldehyde 3-phosphate dehydrogenase. Now this enzyme dehydrogenase, I just want you to focus on the name dehydrogenase. Okay. This enzyme dehydrogenase goes for redox reactions. Always remember, redox reactions. Now, this is the first, you can say, oxidative reaction, because glycolysis is a catabolic reaction, a breaking down reaction where disintegration of glucose is happening. Glyco plus glucose disintegration. So in this very reaction, what you are seeing, what you are looking at is the disintegration of glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate now will get oxidized. So in every catabolic pathway, the golden rule in biochemistry is, when you catabolize something, you have to oxidize. When you anabolize something, means you are creating something, you have to reduce. So here, this is the first oxidation step. So glyceraldehyde 3-phosphate will be oxidized to become 1,3-bisphosphoglycerate. Now you have 1,3-bisphosphoglycerate, that is the truth. Now, here, this is an oxidative product or a product which is the consequence of oxidation. So, something must be reduced side by side. Otherwise, this entire process will be very, very unstable if you look at from the electron perspective. That is why the reactions are redox, because oxidation means removal of electron that must be accepted by something. Otherwise, that electron may get added to some other molecule in the cell, to some lipid or some protein or DNA, and can generate other specific, you can say, pro-oxidants or free radicals, like you can say superoxide, this one, O2 minus, or hydrogen peroxide, or hydroxyl radicals, and they may then directly devastate or destroy, react with DNA, react with lipids. So this OH minus radical reacts with the polyunsaturated fatty acids present in the lipids. It can destroy the plasma membrane, the nuclear membrane, the mitochondrial membrane, and so on. So what you need is that you need that electron to be quenched. Who will quench it? There is a special molecule known as NAD+. Okay, so here two molecules like this. So 2 NAD+, because one for each, will be converted to 2 NADH plus H+. Now this is known as reduction. Reduction has happened, oxidation has happened, they go side by side, that is redox reaction. Still, this reaction is incorrect. If you see properly, how many phosphates do you see in this glyceraldehyde 3-phosphate? There are 2 phosphates, 2 molecules, one molecule having 1 phosphate group each. So there are 2 phosphate groups. How many phosphate molecules do you see in 1,3-bisphosphoglycerate? This one molecule has 1 and 3. At 2 phosphate, at 2 groups, you have bisphosphate, means at 2 different groups. Means it has got at 1 group first carbon, at 3rd carbon. So at 3, you can say at 3rd carbon and 1st carbon, means at 2 sites you have phosphate groups. So 2 phosphate groups per molecule. 2 molecules, so total number of phosphate groups have become 4. Now from where did you get 4 phosphate molecules? Where you have only 2 phosphate molecules. So this is a very special case. What they have used is, or what the cell uses is, PI, 2PI, free PI, which is present in the cytoplasm, in the cytosol. There are PI reserves also. It is not breaking down ATP, from that it is not getting PI. There are PI molecules, okay, which are present in the cytosol itself. And when PI molecules get attached by themselves, not via ATP, there is a very, very distinct thing in biochemistry. Understand it. If from ATP, okay, it is broken down to ADP and PI is used, and then that PI is transferred to some molecule, that molecule is phosphorylated, suppose molecule X is phosphorylated, this will be done by an enzyme known as kinase. But if a simple phosphate group, PI, in the cytosol is attached to a molecule X, okay, this is done by the enzyme phosphorylases. This is a phosphorylase activity. So this dehydrogenase, okay, also has a subtle phosphorylase activity. Okay, therefore the 2 PI molecules are also attached, and then now 2 PI here, 2 P here, phosphate, phosphate, now 4 phosphate, now 4 phosphate. Now the phosphates have been, you can say, balanced completely. So this is what I wanted you to see. In many books, this 2 PI is not given directly. This reaction sequence is completely wrong. Here there are 2, here there are 4. How can you do like that? How can you justify? Because you have to balance this equation in biochemistry. Just like you balance equation in chemistry, we should balance everything in biochemistry as well. So the thing is, there should be 2H plus here. Completely if I write 2 NADH plus 2H plus, because we are dealing with 2 individual molecules, that side 2 PI. So this is the net balanced equation for the first oxidative step, conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate via glyceraldehyde 3-phosphate dehydrogenase, or you can simply say dehydrogenase enzyme. Okay, The cofactor will be Mg2 plus, okay, that is the cofactor for this enzyme also, okay, and this is the first reaction of the payoff phase, the very first reaction. Now if I see, let's draw the structure of 1,3-bisphosphoglycerate so that you remember the structure also. Structures won't be asked in your exam, but I'm just drawing it for you so that you can relate to what's happening inside the cell, because it would be very lame that you just know the name and really not, I do not explain to you the structure. So when you know the structure, you can directly relate to what is happening inside the cell in reality, right? You will get a better picture of it conceptually. So 1,3-bisphosphoglycerate looks like this. You have CO here, and one more O here. To the first carbon, one phosphate, so this is free, one phosphate group here, okay, PO4 3 minus is that P. Then other ways will be looking like, you can say just CHOH, just like how your glyceraldehyde looks, glyceraldehyde 3-phosphate looks. Okay, so glyceraldehyde 3-phosphate is the same as 1,3-bisphosphoglycerate, but at the first position you have, you can say instead of hydrogen, that hydrogen is taken out, okay. In glyceraldehyde, what you have? You have here H. You don't have this one. So here H is taken out and OP is added. Okay, so you have first position phosphate, first carbon, the first carbon, the third carbon phosphate. So this is your 1,3-bisphosphoglycerate. This looks like this molecule. Okay. So after this, now you have 1,3-bisphosphoglycerate. Now we will be having something really fantastic known as substrate level phosphorylation. So 1,3-bisphosphoglycerate, okay, will become 3-phosphoglycerate. Okay, the enzyme here is kinase. I can call it phosphoglycerate kinase, or simply for you I'll call it kinase only. Okay, what it is doing, it is making from 2-ATP reverse. It is making 2-ATP. Now it has made up for the 2-ATPs that were used up. Directly 2 molecules were there. So here 2 molecules. So now 2 ATPs have been produced. Now that debt has been paid. Where in the preparatory phase we used 2 ATP molecules, 1 ATP each, while going for from glucose to glucose 6-phosphate 1 ATP, and then fructose 6-phosphate to fructose 1,6-bisphosphate. So 2 ATPs were used. Now those 2 ATPs have been paid back here. The debt has been cleared. But this phosphorylation is completely different. This ATP generation, okay, just listen to this very, very carefully. Why is it known as substrate level phosphorylation? Substrate level phosphorylation. What is substrate level phosphorylation? Substrate level phosphorylation means there are 2 phosphorylating pathway here, or phosphorylation related pathway. Number 1 I have given you as just about, we will be talking in this chapter only in detail about the electron transport chain, meaning that I think you might have some idea, some crude idea, because Samesh, you learned ARIA is new, but I had taught you Samesh in the NEET class, and also we had discussed that. Just listen to it, this you will just recall it, that in mitochondrial inner membrane, suppose this is the inner membrane of mitochondria, you have multiple transmembrane proteins. Complex 1, complex 2, complex 3, okay. Complex 4, complex 5, which is known as F1 F0 ATPase. This complex 5 is known as F1 F0 ATPase. You might have heard of it. Okay. So this ATPase molecule does what? In the inner membrane space, when the electrons are transferred from NADH to oxygen from complex 1 to 4, you have too much H plus in the system. Here all the H plus are thrown, here very less H plus, here more. So you will have an electrochemical gradient where this pH is lower, this pH is higher because you have more H plus here, and chemical electrical gradient means too much positive charge here, very less positive charge here. So in the end, it will come down its potential energy through F0, F1 F0 ATPase, and that potential energy will be harnessed to phosphorylate ADP and to produce ATP. Okay, so this is the standard way of producing ATP. That's why we say ATP is produced in mitochondria. This entire pathway I will elucidate when I will teach in the next classes about the electron transport chain, which is in your biological transformation chapter in your course only. So the thing is, what happens is, this is not the phosphorylation happening in mitochondria. This glycolysis process happens in cytosol. Okay. So now what is happening is, from this substrate only, I am taking one pi. You can see 1,3-bisphosphoglycerate. So from the first carbon, I am taking one phosphate group out, and that phosphate group is coming with the energy, okay, of the bond breakage, and that energy is used to make the bond between ADP and converted it into ATP. The third triphosphate bond is produced, this gamma phosphate bond, and now ATP is produced. Means ADP has been phosphorylated by a substrate here. Substrate is 1,3-bisphosphoglycerate. It has donated a specific phosphate molecule to an ADP molecule and phosphorylated it to successfully form one ATP molecule. So two molecules formed two ATPs in that process because each donated one phosphate group. Okay, resulting in the formation of two 3-phosphoglycerate molecules. Here you can see the phosphate at the first position carbon is not there, so it is only 3-phosphoglycerate. Now, what does it mean? It means that the phosphorylation has happened not at the mitochondria, not by the or not as a consequence of the electrochemical gradient generated by the electron transport chain, but by the phosphate bond, okay, by the phosphoanhydride bond which was present in the, you can say, or just the phosphate bond, which is a covalent bond which was attached to the first carbon of the 1,3-bisphosphoglycerate. Okay, so that has been donated to the 3-phosphoglycerate molecule, to the ADP molecule to form ATP and form 3-phosphoglycerate in the process. So I told you that whenever this is happening, it is known as kinase. So kinases transfer phosphate from ATP or some molecule or to some molecule or from some molecule to ATP. So both the processes are done by kinases. From ATP, it will transfer a phosphate to some molecule, that is again kinase, or the other way around. From a molecule, it will transfer a phosphate group to phosphorylate ADP and form ATP, again done by a kinase molecule. So therefore, you can say enzyme is kinase, which is protein in nature, and you have formation of ATP. That is why it is known as substrate level phosphorylation. And since this is the first substrate level, you will be having one more. So it is known as first substrate level phosphorylation. A very, very important question in glycolysis, in your biotechnology also, and in your NEET exam also. Which is the first substrate level phosphorylation event in glycolysis? The answer is from 1,3-bisphosphoglycerate to 3-phosphoglycerate. That is the answer in your biotech exam also, in your NEET exam also, or in any other home exam too. Okay. Now we will be talking about the next step. Now we have got 3-phosphoglycerate. So 3-phosphoglycerate looks like what? Just remove the phosphate from the 1,3-bisphosphoglycerate, means you had CO, OP, then CHOH, CH2 then CH2, then directly you can add O here. Then phosphate group. This was 1,3-bis phosphoglycerate. Now just remove this phosphate. From the first position it has been removed. Okay. Now this will be minus now. So this is your 3-phosphoglycerate. This is the 3-phosphoglycerate molecule. Okay. This is the structure of 3-phosphoglycerate. If you had a phosphate group here attached, that is 1,3-bis phosphoglycerate, but that has been removed, so it is a minus sign here. One electron is remaining extra, that is 3-phosphoglycerate. Now, the next step will be to convert 3-phosphoglycerate, okay, to another molecule known as 2-phosphoglycerate, means transferring the phosphate from the third position or the third carbon to the second carbon. So that is done by a very specific enzyme known as mutase. Mutases, they do what? They transfer the phosphate group from one carbon of one compound to another carbon of the same compound. The same phosphate group they will, you can say, transfer. I'll show it to you. What do I mean by this? What do I mean is, you will get the molecule COO, O minus, okay. Now, here is the, you can say, second carbon, so CH. Here O, here P. Now phosphate has come here, okay, and then this side will be CH2OH. So from this molecule to this molecule. In this molecule, what was the thing? On the third carbon, you are having the phosphate 1, 2, 3. Here on the second carbon only you have the phosphate. So phosphate has been put here, okay, and this thing has been transferred there. That's all. Okay, this entire group, this group has been transferred here, okay, and this group has been transferred there. That's all that it has been done. You can see OH, you can see here, and CH2 was already there, OH here, and this entire thing was, it has been put here. You can see this has been transferred here and this has been transferred here, and what you get is 2-phosphoglycerate. This molecule is 2-phosphoglycerate. Just check this. That is why I am showing it to you, the structures. Otherwise it will become very, you can say, baffling for you that what is going on. Okay. This is what's going on. This is done by the enzyme known as mutase. Okay. So mutases, they do what? They transfer the phosphates. Okay. They transfer the phosphate from one molecule's one carbon to the same molecule's other carbon, because it was in the attached to the third carbon, or the same phosphate is attached to the second carbon. That's the only job that the phosphates are able to do, are able to catalyze. Okay. Now next is, I can write this equation for you. 3-phosphoglycerate becoming, you can say, 2-phosphoglycerate by phosphoglycerate mutase enzyme. Simply mutase enzyme, 2-phosphoglycerate. This is what you can think of. Now next step in glycolysis, okay, if you see, will be formation of, you can say, from 2-phosphoglycerate you will be forming phosphoenol pyruvate. Okay, there will be an enzyme known as enolase which will take out H2O from this specific, you can say, a molecule, and you will be forming phosphoenol pyruvate. So from 2-phosphoglycerate to 2-phosphoenol pyruvate, 3 carbon going on, so it's still 6 carbon, 3 plus 3 6 for each one. Okay, 3 carbon compound. So 3 carbon compound. So we started from glucose, so 6 carbons, Here 2 3 carbon means we are maintaining the stoichiometry of 3 carbon. Now in the end 2 phosphoglycerate will be changed to or will undergo a reaction by this specific enzyme known as enolase. One H2O will come out and it will become 2-phosphoenol pyruvate. Phosphoenol pyruvate. Now, just I'll again draw both the structures for you. Let's draw 2-phosphoglycerate. Okay, C. Then second carbon will have the phosphate group. Okay. Third carbon will be having this is 2-phosphoglycerate, okay, molecule. Now it will be changed to reversible reaction phosphoenol pyruvate. Remember, I have to take out one H2O. So I'll take out this H from here. Okay. Cause this double bond to be made. Okay. And once double bond is made, only two hands are free. This is phosphoenol pyruvate. Now see, OH gone, this H gone. So H2O is released, right? This OH is gone and this H is gone. 2H and 1O, H2O is released. So this is your phosphoenol pyruvate. Look at this molecule carefully. That's why learning why structure helps. And this P is not P. This P is wherever I am drawing P in a circle, that is a phosphate group. Phosphate groups are like this. PO4 3-. So the thing is, this is known as phosphoenol pyruvate. You can now simply understand, comprehend and decipher what is going on. It is now pretty easy via structural biochemistry that what is happening. Now if I just explain to you 2-phosphoglycerate, then 2-phosphoenol pyruvate means phosphoglycerate is being converted to, or 2 molecules of 2-phosphoglycerates are being converted to 2 molecules of 2-phosphoenol pyruvates. You will think what is happening, H2O, where it is, from where it is coming out. So this is the entire thing is in front of you now. Mechanistic detail. One OH, one H went out, and now you have a specific phosphoenol pyruvate molecule synthesis in front of you, right? Now, after this phosphoenol pyruvate has been produced, so you have got 2 phosphoenol pyruvates. Now both these molecules have one phosphate group, right? Now what you can go for is second substrate level phosphorylation. You can lose that phosphate too and produce a simple molecule known as pyruvate via a pyruvate kinase. So pyruvate kinase, or a simple kinase I can say. Pyruvate ADP to ATP. 2 molecules are there, they will be forming 2. So if you see the structure, okay, I've already shown you the structure, I'll draw it again. The phosphoenol pyruvate structure. See here. So this is your phosphoenol pyruvate, and now this phosphate needs to be removed. Okay, so what you will get is, you can put a double bond here, there also. Okay, very easy. You can put a double bond there, and when you put a double bond there, it becomes a very specific double bond. And one more, you can say, what you can do is, you can put one more H. Okay. So what will happen is I can put a double bond here. This phosphate gone to attach to the ATP. Okay, and then I can put C H H H CH3. Okay, so this will be my pyruvic acid or pyruvate. In biochemistry, acids are known by the name 8. 8 or ic acid is same. Pyruvic acid, pyruvate, lactate or lactic acid, amino acid, aspartate or aspartic acid, okay, glutamate or glutamic acid. The thing is, now that phosphate group has been lost, and it's being not lost, it is being, this specific bond has a lot of energy. So that energy is being transferred, and it is being used to phosphorylate ADP by this kinase and produce ATP. It is not happening in mitochondria, not happening via the electrochemical gradient in the inner mitochondrial membrane within the intermembrane space. The gradient is present, so it is happening in the cytosol from one substrate. This is a substrate to, you can say, transferring phosphate group to ADP. Okay, so that is known as substrate level phosphorylation. It is the second substrate level phosphorylation event. Second substrate level phosphorylation event. Now it is in the form of the formation of formation of pyruvate. Okay, now you have been able to form pyruvate. Okay, so now it's quite easy for you now how to comprehend this entire process of glycolysis. Okay, now in some portions or in some specific pathways of glycolysis, just like we converted, you can say, fructose 6 phosphate to fructose 6 bisphosphate. So I have put a single arrow here. Also see, I have put a single arrow in the last process. Some processes I have put double arrow. Double arrow means where the del G is little bit positive or slightly negative. Mostly it is little bit positive, means the reaction should not be happening, but since the product concentration is, you can say, it is going towards the product concentration, reactant concentration is too high, going towards the product concentration, so the reaction is happening. Okay, so if you have studied thermodynamics in chemistry, you will realize that for a spontaneous reaction to happen, the del S should be positive and del G should be negative. Okay, so that is the reason why in some cases single arrow has been put, that is del G is highly negative, it is an irreversible reaction, and if it is little bit positive, okay, then it will be, you can say, a specific, you can say, reversible reaction. Like you have, suppose the aldolase, let's take the example of from fructose 1,6 bisphosphate, I formed dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, in that reaction, fructose 1,6 bisphosphate was converted to, first of all, glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate, right? So this was the reaction that we started off with. Just to give a perspective of the energies. The energy here, the enzyme is aldolase. So this is a reversible reaction I have written. Why? Because the del G for this reaction is positive 23, del G naught, okay, at equilibrium HTP conditions is 23.8 kilojoule per mole positive. Okay, so what you have is the aldolases. Okay, the aldolases are of two types. There are class 1 aldolases which are found in animals and plants, and there are class 2 aldolases which are produced in, or which are found in bacteria and fungi. Okay, so what you have is the aldolase reaction has a strongly positive standard free energy change in the direction of, you can say, fructose 1,6 bisphosphate cleavage. Okay, but at the lower concentration of reaction present in the cells, the actual free energy change is very small, okay, and the aldolase reaction is readily reversible. If you have a lot of fructose 1,6-bisphosphate, okay, if you have a lot of fructose 1,6-bisphosphate, it will never convert, the aldolase enzyme can never convert a huge amount of fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. But in the cell you have picomolar, nanomolar concentration of fructose 1,6-bisphosphate. So because of this very little or minuscule concentration, the standard free energy in reality in the cell del G is not that highly positive, it is little bit positive, so it can proceed in the, you can say, this direction, and the aldolase can then break it down because of its very low concentration inside the cell. If it was present in high concentration, then you would have never been able to go in this direction. It will always favor this direction because the del G of this direction is positive. You cannot have positive, okay, in this direction, or it is highly positive. So if you want a reaction to happen, then the del G must be negative, and if it is highly negative, then it becomes irreversible reaction, right? So wherever you have seen double arrow means del G is little bit positive, but it is going on because the concentration of the substrates is very low, so it is being able to compensate that thing, okay. So that is what I wanted you to understand. So the last part of this glycolysis that I want you to know, okay, is the specificities when it comes to formation of pyruvate, okay, formation of pyruvate. So in most of the enzymes like mutase and all, they all use Mg2+ as cofactor. That is what one thing I'll make clear, okay. And next thing is, if you see, okay, the pyruvate molecule, the pyruvate kinase. So pyruvate kinase uses Mg2+, okay, this is a very deep, neat question. Pyruvate kinase, when phosphoenol pyruvate is being converted to pyruvic acid or pyruvate, here you have kinase molecule, and this kinase molecule you are making ADP, so this is 2, this is 2, so 2 ATP to 2 ATP. So this kinase molecule is also using Mg2+ or K+, okay. It can also use K+ as the cofactor, always remember. And once pyruvate is produced, okay, once pyruvate is produced, what happens is, it is produced in the enol form. If you have studied tautomerism, I'll just draw it for you. It is produced in the enol form. Just look here. You can say OH, CH2, this side will be, this is pyruvate, this is the enol tautomer. If you study tautomerism, you will be able to comprehend, otherwise also you will be able to comprehend. Now, what happens is, this gets tautomerized, okay, to the keto form. Keto form is an isomer only. What happens is, C, changing of the oxygen. Oxygen will be, you can say now, double bonded this oxygen, okay. This will be CS3 now, okay, this will be CS3, and then it is the same, then it is the same. So this is known as the keto form. So what happens is, the pyruvate first appears in the enol form, which tautomerizes rapidly and non-enzymatically. There is no enzyme needed, okay, in its keto form, which is predominant at our physiological pH of 7.35 to 7.45, okay, that pH is necessary for that, okay. So now this overall reaction has a very large negative del G, okay, of about, you can say, 61.9 kilojoule per mole, okay, because you can say the formation of phosphate bond, that energy was 30.5 and the rest 31.4 was due to the, you can say this keto to, you can say enol transformation. Okay, so or you can directly think of that half of the energy released by phosphoenol pyruvate hydrolysis, okay, is conserved in the formation of the phosphoanhydride bond. Okay, phosphoanhydride bond is conserved, so 30.5 kilojoule per mole is conserved in it, and the rest 31.4 kilojoule per mole constitutes the large driving force which pushes the reaction towards ATP synthesis. Means that this pyruvate still has a lot of energy left. It contains almost 30.5 kilojoule per mole of energy, 30.5 kilojoule per mole of energy in itself, okay, which will be used up in Krebs cycle, and the real del G is, you can say, minus 31.4 kilojoule per mole. But the actual practical will be what? You can say 30 plus 31 is 61. So it will be 61.9 kilojoule per mole, right? Actual one minus 61.9. So that will be the actual, but this is stored, so this is not counted. This is what is we get after the ATP is formed from ADP. ADP gives rise to ATP phosphorylation. So this is what we get from that specific reaction. Okay. So this is what happens during that process, and this enol to keto conversion helps to further stabilize pyruvate and hold on to this 30.5 kilojoule per mole energy, because it is more stable at its keto tautomer at physiological pH of 7.35 to 7.45. Okay, so that is what this entire process of glycolysis is all about. So if you have any, you can say, doubts, you can clarify it on my personal number. Okay, I hope you are able to comprehend the lecture, and if you have any doubts, you can ask me at any time, and then we'll be meeting you again in the next class. Okay then, thanks a lot. You toddler.